Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different aspects the of the molecules in the course molecular biology. So far what we have discussed about the cellular structures in the initial modules, then followed by we have discussed about the different types of biomolecules. So we have discussed about the DNA, RNA, proteins, and then we also discussed about the enzymes. And following that, we have also discussed about the different types of cellular processes. So we have discussed about the central dogma of molecular biology, and then we have seen the detailed discussion about the different types of processes what is occurring within the central dogma of molecular biology. So we have discussed about the replications, transcription and translations. And with this kind of discussion, we understood that the molecular biology has developed the different types of basic principles on which you can be able to develop the different types of techniques, and these techniques can be used even to explore the aspects of the molecular biology, or they can also be used for different types of applications. So, in today's lecture, we are going to discuss about some of the important molecular techniques and how these molecular technique can be used for answering some of the basic questions related to the cellular physiology or other aspects related to the molecular biology. So, when we talk about the molecular biology is all about the molecules, right, and these molecules interact due to a specific requirements. I am sure you can recall when we were discussing about the proteins. We said that the protein is interacting with its substrates and protein is interacting with the other proteins due to the recognition of the complementary regions, due to the specific three-dimensional requirements, geometric constraints, stereospecificity and other kinds of requirements. And because of these kind of specific requirements, the molecular biology or the interaction between the molecules can lead to the development of different types of techniques. So, when we talk about the molecular technique, can be of many types. Okay, molecular technique where you are actually going to amplify a particular DNA or other kinds of regions. So, it can be a amplification technique. Within this amplification technique, you can have the different types of amplification techniques such as the polymerase chain reactions, you can have the real time PCR, or you can also have the RT-PCR. Okay, so this is I will write real time PCR and this is the RT-PCR or reverse transcriptase PCR. And all of these amplification techniques are having the utility in terms of the amplifying a specific region of the genome, so that you can be able to get the genome fragment, and these fragments can be used for multiple for the downstream cloning applications and all that. So, that anyway we are going to discuss in detail when we are going to take up the molecular cloning. Then apart from that, you can also have the different types of the blotting techniques. So, within the blotting techniques, you can have the techniques which is called as southern blotting. You can have the northern blotting, and you can also have the western blotting, and you can also have the eastern blotting. Okay. So, most of these blotting techniques where you are actually going to use the hybridization techniques and you are actually going to blot the molecule of your interest onto a support system, and then you are going to detect that with the help of the different types of the analytical reagents. For example, in the case of southern blotting, you are going to use as the DNA as a probe, and northern blotting also you are going to use the DNA as a probe, and then western blotting you are going to use the different types of antibodies and so on. Apart from that, you can also have the molecular techniques like where you are actually going to have the sequencing technique. So, you can actually be able to do the sequencing of the DNA and protein also. So, you can have the sequencing of the DNA, or you can also have the sequencing of the proteins, and that can be useful in terms of the identification of the that particular region or accuracy of the cloning and other kinds of things. And there are many techniques. For example, you can have the RFLP, you can have the you can have the small molecular polymorphisms and all those kind of things. So, molecular technique is a very vast subject, and there could be a MOOCs course on that actually. So, what we are going to do is, within this molecular technique topic, what we are discussing, we are going to discuss about the amplification techniques, because they also going to have the downstream applications into the molecular cloning, and then we are also going to discuss about the blotting techniques. So, in current module we are discussing about, we are going to discuss about the blotting techniques. So, within the blotting technique we are going to discuss about southern blotting, northern blotting and western blotting, and in the next module we are going to discuss about the amplification techniques such as the PCR, RT-PCR and real time PCR. And all of these techniques will help you to understand the application part, that also we are going to discuss at the end of this particular course. So, as far as the blotting technique is concerned, the blotting techniques are being categorized based on the target molecules. So, for example, as I said, we have the 4 different types of blotting technique. We can have the southern blotting, we can have the northern blotting, we can have the western blotting, and we can also have the eastern blotting. And the southern blotting, you are going to use the DNA as the target molecule. So, you are going to identify the DNA. If DNA is a molecule, then it is going to be called as the southern blotting. If you are going to detect the RNA, then it is going to be called as the northern blotting, and if it is the protein, then it is going to be called as the western blotting, and if it is a carbohydrate, then it is going to be called as the eastern blotting. Okay. Although eastern blotting is not very popular, but since they were having the southern, northern and western blotting, the people have, whatever the technique is using for detecting the carbohydrate onto the nitrocellulose membrane or other kinds of support system, that is being called as the eastern blotting. So, eastern blotting is not very popular, and that is why we are not discussing about the eastern blotting in this particular course. So, we will start with the southern blotting, then we are going to discuss about the northern blotting, then we are going to discuss about the western blotting, and then lastly, and so when we discuss about these techniques, we are going to talk about the basic principle of the technique, and then we are also going to talk about the underlying mechanisms, and we are going to talk about how to perform this particular technique. So, let us start with the southern blotting. So, southern blotting is a 6-step blotting technique, and remember that in all the blotting techniques you are going to blot the material onto a support system such as the nitrocellulose membrane or the PVDF membranes, and then you are going to perform the reactions on that particular support system. So, the first step is, for example, so northern blotting is for DNA, right, and the purpose of northern blotting is to detect a fragment of DNA into a particular genome or a genomic library. So, the first step is that you are going to have the genomic DNA of that particular organisms, and you are going to isolate the genomic DNA from that organism. Then in the second step, you are actually going to do the digest the genome, because genome is very long, right. So, you cannot analyze that where the actually the probe is binding, right. So, what you can do is you can digest the genome, so that you are going to have the different types of fragments. And then in the third step, you are actually going to separate these DNA fragments onto a suitable agarose gel, right. So, you are going to use that suitable agarose percentage of the agarose gel, and then you are actually going to analyze that, and that is actually going to give you the separate fragments. Then in the step 4, these DNA fragments are going to be transferred onto the nitrocellulose membrane with the help of the different types of transfer techniques. So, you can use the manual method, or you can use the high vacuum method as well. Once it is being done, then you are actually going to prepare a probe, and you are going to prepare a radio labeled probe. Probe is a small fragment of DNA that you are actually going to use to detect this specific DNA into the genomic DNA. So, you are going to use the hybridization with the radio labeled probe, and then in the step 7 you are actually going to do a washing, and then you are going to do the autoradiogram. And at the end of the autoradiogram you are actually going to get the result, and that result can be interpreted in terms of how many different DNA fragment are present into the genome and how many different copies. So, basically with the help of the Southern blotting you can be able to say whether this particular DNA fragment is present in that particular genome or not. And the second information what you can also extract is what will be the copy number, how many fragments or how many different gene of that particular fragment is present in that particular genome. So, it is actually going to tell you the location of that DNA fragment, and it also going to tell you the information about the copy number, right. So, so far we have not discussed about the copy number, but I think we'll going to discuss when there will be a molecular cloning and other kinds of events. So, the first step is that you are actually going to start with the genomic DNA isolations. So, genomic DNA isolation is also a step 1 in the genomic DNA isolation. So, genomic DNA isolation is also a multi-step process. In that multi-step process, the first step is that you are going to do the lysis of the cell with the help of the detergent containing the lysis buffer. Now, at this stage you can have the two different types of sources. You can have the either the cell, or you can actually have the tissue. For example, let us take an example of the liver. So, for example, if you have a liver from where you want to extract the genomic DNA, and then you want to ask the question how many copies of that particular gene is present into the liver genome, right. Cell, we can take an example of the cell also. So, for example, hepatocyte. So, whether you use the tissue or whether you use the cell, the ultimately is that if you are trying with the tissue, then it has to be fragmented and into such a case that it should reach to a cellular level. So, tissue is made up of the different types of cells. So, what you are going to do is you are going to use the enzymes which actually can dismantle the intercellular spaces. For example, you can use an enzyme which is called as collagenase. So, if you treat the liver with collagenase, collagenase is an enzyme which actually degrades the collagen fibers, and you know that the most of the cells are stick to each other because they have a matrix in between, and this matrix is made up of the collagen or the collagen like material. So, in the liver you have the collagen like material which is actually sticking these cells. So, if you treat them with the collagenase, what you are going to get is you are going to get the cellular suspension or single cell suspension, and this single cell suspension can be used for the further downstream. So, single suspension means from the liver you got the single cell suspension, and now you can actually be able to treat them with the help of the detergent. So, whether you are using with the single cell or whether you are using the tissue, at the end you are going to get the cells, and then you are actually going to treat them with the detergent. And once you do with the detergent treatment, the cell is actually going to break open, then it is actually going to release the cellular content. And what is there in the cellular content? The cellular content is actually going to contain the cellular proteins, it is going to have the RNAs, it is actually going to have the lipids, and it also going to have the proteins, and it also going to have the genomic DNA. So, now from the cellular content, your job is that you want to isolate the DNA, but you want to get rid of, you do not want that. So, you want to get rid of the protein, you want to get rid of the lipids, you want to get rid of the other materials, right. So, that is what you are going to achieve in the subsequent purification steps. Now, in the step 2, you are actually going to incubate the cells with the digestion buffer containing the proteinase K, SDS to release the genomic DNA from the DNA protein complexes. Remember, when we were discussing about the genetic material, we said that in the eukaryotic system, the genetic material is associated with the protein in the form of the nucleosome, and these nucleosomes are even forming the higher order organizations, right, such as the chromosomes and other kinds of things. So, the first step is that you want to isolate or you want to dislodge the protein part, so that the DNA would be free, and then you can be able to get the purification of the DNA from the cellular material. So, that for that what you are going to do is you are going to incubate the cellular content or the lysed cells with the digestion buffer, which contains the proteinase K and SDS. So, in the presence of SDS, the protein is actually going to get denatured, right, and because the protein is going to denature, it is actually going to lose its affinity for the DNA. On the other hand, the proteinase K is a protease, right. So, it is actually going to degrade the protein into the peptide or peptide fragments, right. So, ultimately what you are going to get is you are going to get, and peptide fragment can be digested. So, if you continue the digestion, it is actually going to get converted into amino acids, right, because the proteinase K is a non-specific protease. So, it is actually going to start chewing up the proteins, and ultimately what you are going to get is you are going to get the DNA, which is separated from the DNA protein complexes, plus you are going to have the protein, which you are going to get from the cellular content, and then you are also going to have the lipids, right, because lipid is also present in the cellular material, and some minor component of, like for example, you can also have the RNA, right. So, now what you have is you have a mixture of DNA, protein, lipid and RNA, and your target molecule is the DNA. So, you can actually devise a strategy so that you can be able to purify the DNA, but get rid of the protein, lipid and RNA species. So, that you can do sequentially by removing the protein, lipid and RNA, and that is how you are going to get, at the end you are going to get the purified DNA. So, in the step 3, you are actually going to isolate the genomic DNA by the absolute alcohol precipitation. So, what you are going to do is you are going to add the absolute alcohol to the thing, and it is actually going to precipitate the genomic DNA. But when it is going to precipitate the genomic DNA, so it is going to give you the genomic DNA, but it is also going to have the small amount of proteins, it is also going to have the small amount of lipids, and it also going to have the small amount of RNA, especially the messenger RNA. So, these has to be get rid of, right, these has to be get rid of. So, in the next step what you are going to do is you are going to do the purification of the genomic DNA with the help of the phenol chloroform isoamyl mixture. And when you mix the phenol chloroform, one mixture has two phases, right. You are going to have the aqueous phase and you are going to have the organic phase. See, here you have the two phases, you have the chloroform phase, which is going to be the part of the organic phase, right. And you know that when we are going to do a fractionation with the solvents, the molecules will actually go to their respective phase, right. For example, if there is a organic material, then it actually going to prefer to the organic phase, and if it is a polar molecules, then it will go to the polar environment, right. And as a result, what will happen is that in a Eppendorf, you are going to have the two phases, you are going to have the organic phase and you are going to have the aqueous phase. So, this is going to be aqueous phase and this is going to be organic phase. So, all the molecules which are soluble into the organic solvents, such as lipids, such as the other kinds of molecules, they will be present in this, whereas the DNA and RNA is going to be present into the aqueous phase. So, in this step, the phenol is actually going to denature the remaining proteins and keep the protein into the organic phase. So, organic phase is actually going to take care of the lipids. So, it is actually going to remove the lipids, it is going to remove the protein, because protein is going to be denatured by the phenol, and as a result the protein is also going to be present into the organic phase. Whereas, in the aqueous phase, you are going to have the DNA and you are also going to have the RNA, because RNA is also going to be polar in nature. Now, next step is that you want the DNA, but you do not want the RNA. So, okay. So, then you are going to do a further purifications. So, first you are going to do isolation of the genomic DNA what is present in the aqueous phase, and it will again precipitated with the absolute alcohol. So, if you do the reprecipitations, it is actually going to precipitate the DNA, and then it is also going to have the small amount of RNA, and that you can actually be get rid of by putting the things into the RNAs. So, if you incubate this mixture with the RNAs, then RNAs is actually going to chew up the RNA, and it is actually going to destroy the RNA, and then ultimately your genomic DNA is going to be isolated. So, this is what exactly is being depicted in this particular picture. First step, you are going to do the lysis. Second step, you are going to do the enzymatic digestion. The third step, you are going to precipitate the genomic DNA with the help of the absolute alcohol. But when you are going to precipitate along with the genomic DNA, you are also going to get the lipids and the protein part, and that you are supposed to get rid of with the help of a purification step. So, in that purification step, you are going to add the phenol chloroform mixture, and when you add the phenol chloroform mixture, the phenol is actually going to denature the proteins, whereas the chloroform is actually going to remove the lipid part. And when the phenol is going to denature the protein, they also will be come into the organic phase. So, when you collect the aqueous phase, this aqueous phase is actually going to contain the DNA and as well as RNA, and then from this aqueous phase you can precipitate the alcohol with the help of absolute alcohol. You can again precipitate the DNA, and then you can treat this DNA with the small amount of RNAs, so that you can get rid of the RNA part, and then ultimately you are going to get the pure mammalian genomic DNA. Now, when you run the mammalian genomic DNA onto agarose gel, what you are going to see is that you are going to see an intact band which is going to be of high molecular weight. Okay. So, it is going to be of high molecular weight rather than a low molecular weight, and this is going to be indication that you have isolated the genomic DNA. If it is not a genomic DNA, or if it is not an intact genomic DNA, then in that case you are going to see a band, but below to that band you are going to see a streak, okay, or you are going to see a smear. So, that smear is actually an indication that you have broken the genomic DNA, and you are actually going to generate the, or you have degraded the genomic DNA. So, that is not good for the further analysis of into the sudden blotting. Now, the next step is step 2. You are going to have the generation of the suitable size fragments, because genomic DNA is very long. For example, when we were talking about the genetic material in the eukaryotes, we said that in the humans the length of the genomic DNA is approximately around 1 meter, right. So, this kind of lengthy genomic DNA cannot be analyzed for for probing purposes. So, what you want is that you digest that genomic DNA, so that you are going to have the small fragment, and then depending upon the binding of the probe, it will actually going to say that okay, if the probe is binding to the multiple location, that means that many number of copies are of that particular fragment is present into the genomic DNA. So, for that purpose the genomic DNA has to be fragmented with a suitable system, so that you are going to have not very big fragments, not very small fragment, but should be a suitable size fragments, and that you can be able to achieve by the two different methods. First, you are going to do a restriction digestions. So, genomic DNA can be digested with a frequent DNA cutting enzymes such as EcoR1, BAMH1 or SAW3A to generate the random sizes of their DNA fragments. The criteria to choose a restriction enzyme or pair of restriction enzyme in such a way, so that the reasonable size DNA fragment will be generated. As fragments are randomly generated and are relatively big enough, it is likely that each and every genomic sequence is represented into the pool. So, the purpose is that you should not have very small fragments, you should not have very big fragment, it should be a relatively reasonable size, so that each and every genomic sequence should be presented, which means it should be of such a size that a single gene can be present into that each fragment, and because of that each fragment is actually going to represent the individual genes, okay. And because of that, if you are going to see that the probe is binding at multiple location, that means your gene is having that many copies present within the genome. Then the second method is the mechanical sharing. So, mechanical sharing genomic DNA can be fragmented using the mechanical sharing. Mechanical sharing method, you can use the vertex, you can use the sonication and so on. But all these mechanical methods are need to be optimized very nicely, so that you should not have very large fragments, because large fragment have no use, because it is going to have the representation of the multiple genes, right. So, because of that it may actually going to give you the misguide. Now, in the how you are going to perform the restriction digestion, right. So, in the step 2, restriction digestion can be done with the help of the restriction enzyme. So, what you need to do is you digest the 10 to 20 microgram of genomic DNA with the EcoR1 overnight in the presence of appropriate buffer and BSA. So, when you are going to use any enzyme, you require a suitable buffer and the other kinds of additives such as BSA and magnesium and all of the kind of things. Then take out the small aliquot from the restriction digestion reaction and check it onto the 0.8% agarose gel. Appearance of the smear with a small band, visible band, indicate the complete digestion of the genomic DNA and the suitability of the sample for the Southern blotting. This means, if I am going to run the genomic the restriction digestion in the control sample where I have not added a restriction enzyme, I am going to see an intact band of the genomic DNA, just now what I have shown you into the agarose gel. But if the cut fragment or restriction enzyme, wherever restriction I have, I am going to see a fragment, and then I am going to see a complete smear kind of situation, which means I am going to see a white band with the discrete band also. So, that means this is actually being done correctly, and all these fragments are suitable for the Southern blotting, and the step 2 is already been done. Now, you can move on to the step 3, that means you are actually going to separate these DNA onto a suitable gel system. So, the step 3 is the separation of DNA onto the agarose gel. So, separation of the DNA onto the agarose gel. Agarose gel electrophoresis is a standard gel to resolve the DNA. As a standard practice, the agarose used for the Southern blotting is of ultra pure quality to avoid the contaminants affecting the migration of DNA. Observe the gel in the transilluminator and record the pattern in a gel documentation system. So, this is very easy to say, but it is not easy, right. You are supposed to prepare an agarose gel, then you are supposed to prepare the sample, and then you are supposed to run it onto the agarose gel, and then only you are going to see the pattern. So, for demo purposes, I would like to take you to my lab where the students are actually going to show you the how to perform the agarose gel electrophoresis and how you can be able to observe the gel under the transilluminator and how you can be able to do the documentations.
Hello everyone, in this video we will be demonstrating how to set up a PCR reaction and analyze the results using agarose gel electrophoresis. PCR or polymerase chain reaction is a widely used molecular biology technique to amplify a particular segment of DNA. It is also employed in biomedical research and forensic medicine. The main application of this polymerase chain reaction is cloning. To set up a PCR reaction, we need template DNA, site-specific primers, DNTP mix, nucleus-free water, and Taq polymerase. For a 50 microliter reaction, in a typical concentrations of 10 to 100 nanograms of template DNA used and 5 picomoles of each primer will be used. This is a earlier version of thermal cycler which contains display unit where we can observe the parameters and change the parameters. This is a hard shield. This is sample holder, and inside there is a Peltier system which can maintain the temperature fluctuations. For setting up a PCR reaction, initial denaturation at 95 degrees Celsius 3 minutes, and these steps we will use 30 repeats, where initial denaturation will be 30 seconds and annealing at extension time should be given 1 minute per kb. And here final extension should be given 10 minutes and hold at 4 degrees Celsius . Once the PCR reaction is completed, we have to analyze the results for amplification. For that we need agarose and TAE buffer. First we have to weigh agarose and mix with the TAE buffer. It will not dissolve easily, so we have to heat it in microwave oven until it gets dissolved. Now agarose got dissolved in TAE buffer. We have to let it cool down up to 50 degrees Celsius. Now before pouring we have to add ethidium bromide for detection purpose. Now the gel got solidified. We have to take out the gel and keep it in the electrophoretic apparatus. We have to gently remove the comb loose the knobs and keep the gel in the apparatus. Make sure that the buffer is submerged the gel. We have to fill the remaining part with 1x TAE buffer. Generally for analyzing the DNA samples we will use agarose gel electrophoresis. This is the power pack and this is the electrophoretic apparatus. This is a negative electrode and this is a positive electrode. We can change the voltage from here. For loading up sample, we have to mix PCR reaction mixture with 5x loading time. The loading is over. We have to cover the electrophoretic apparatus with the lid, and we have to adjust the voltage. Then start run. After the agar gel electrophoresis, we have to visualize the amplified product. This is the Chemiduct MP, where we are going to visualize the amplified product. Now we have to keep the gel then close the thing. We have to select here application, nucleic acids, ethidium bromide, exposure, optimal exposure, or we can select manual also. Then we will acquire the images. Now we can find here, this is the DNA ladder, this is the PCR amplified product. We can transform or save this image into JPG or. In this video we have discussed how to set up a PCR reaction and how to analyze the PCR result. We have also shown how to use a thermal cycler and what are the components of thermal cycler. So during all this process we have to take some precautions to get better results. Like all the time you have to keep polymerase enzymes and your primers on ice, and other thing we have to remember is while running the agar gel electrophoresis, always wear the gloves to prevent any contamination with the ethidium bromide. So ethidium bromide is a carcinogen, so it is not very likely to cause any cancer, but we have to make sure that we are avoiding this kind of touching with the ethidium bromide.
Now let's move on to the step 4. So in the step 4, you are going to transfer the DNA from the gel to the nitrocellulose membrane. So you have two different types of method, either you have capillary transfer method or you can actually have the vacuum manifold. So in the capillary transfer method, you are going to soak the gel into the 0.2 normal HCl for 10 minutes with shaking at room temperature. This step depurinate the DNA and facilitate the transfer of larger DNA fragment, and this step can be omitted if the fragmented DNA is of smaller size. So this is a very important step which is going to be required only when you have a large fragment, so that you can actually be able to soak the gel into the 0.2 normal HCl, so that all the purines are going to be depurinated, or all the nucleotides are going to be depurinated, and that actually is going to increase the efficiency of the transfer. Then you can remove the depurination solution, wash the gel with the double distilled water and submerge the gel into the alkaline solution for 30 minutes at room temperature with the gentle check-in. Alkaline solution is actually going to denature the DNA, and that is how the DNA is going to be not present as a double stranded DNA, it is going to be present as a single stranded DNA. Then you remove the denaturating solution, wash the gel with the double distilled water, submerge the gel into the neutralization solution at 30 minutes for room temperature with gentle shaking. This step will neutralize the gel without denaturating the DNA. Then you pour off the neutralization solution and incubate the gel into the 10x SSC buffer for 30 minutes with the gentle shaking. If you do not know the composition of the 10x SSC, then you can actually be able to refer any standard molecular biology book. Okay. And these are the standard buffers which are very for which the recipe and how to prepare these buffers can be found. Then the preparation of the nitrocellulose membrane for the transfer. So, cut the piece of the nylon membrane to the size of agarose gel, mark the nylon membrane by cutting on the one corner, wash the nylon membrane with the double distilled water and then soak the membrane in a transfer buffer. So, what you are going to do is you are going to have the nitrocellulose membrane which is of the same size as your agarose gel block, right, and then you are going to cut on one side. You are going to cut with the scissor, so that you know that this is the side on which I was having the lane number 1, lane number 2, lane number 3. Okay, so that it should you are going to maintain a polarity into the membrane, so that you can be able to identify that the site at which I have cut is actually the site of where I was having the sample number 1. Okay. Otherwise, this is going to be a problem, because this after the transfer is done, then you do not know which one is sample number 1 and which one is sample number 3. Then you cut the two pieces of 3 mm Wattman filter paper slightly bigger to the side and wet the filter paper with the transfer buffer. And this is the apparatus, or this is the setup what you are going to use for the transfer, okay, where you are going to have the agarose gel, you are going to have the transfer membrane, you are going to have the blotting sheets, and then you are going to have the weight. And below to that you are going to have the transfer tank. In this transfer tank you are going to have the transfer buffer, and on this transfer buffer is going to be connected to the transfer tank with the help of the wicks, right. So, you see this is the wicks, right, which are going from top, and so you are going to set up the blotting tray. So, add 500 to 8 ml the transfer buffer, which is 10x CSC, in the transfer tray. On a glass solid support, place a piece of non-woven wiper with the size of the solid support to serve as a wick. So, what you are going to do is, you are suppose this is the glass support. So, what you are going to do is you are going to put a wick, so that it is actually going to go like this, and on this you are actually going to put your agarose block, and then on top of agarose block you are going to put the transfer membrane, that is the nitrocellulose membrane, and then on top of that you are going to put the blotting sheets, and then ultimately you are going to put a weight. And then what will happen is that here you are actually going to have the transfer buffer. So, the transfer buffer will go from this side, and then it is actually going to go to these blotting sheets. So, there will be a suction pressure which is actually going to transfer the DNA what is present into your agarose block onto the nitrocellulose membrane. So, dip the both end of the wick in the transfer buffer, wet the wick and remove the trapped air bubble by rolling with the glass rod. Then you place the gel, and so then you place the gel on top of the wick and remove the bubble by rolling with the glass rods. Then you overlay the nylon membrane onto the gel and carefully remove the trapped air bubble by rolling the rod, right. So, you are going to have the your agarose block, right, and then on top of this you are going to put your the nitrocellulose membrane, and on top of this you are going to put the blotting sheets. You are going to put the lot of blotting sheets, right, and then on top of this you are going to keep a weight, right, so that it should remain in touch with each other. And below to this you are going to have the wicks, right, which are being submerged into a transfer buffer. So, here you are going to have the transfer buffer, and while you are doing all this, all this has to be handled with the utmost care, that you are actually going to wear the gloves, because you know that human body is also going to secrete the biological fluid, and this biological fluid may contain some of the DNA and RNA. So, that is how you can be able to contaminate. On the other hand, the human body is not sterile, so it may also have the bacteria and other kinds of infectious organisms or non-infectious organisms, So, that also may contribute in terms of their with their own DNA, and that may actually increase the background. So, it is very important that you should perform all these reactions with the help of utmost care, utmost cleanliness, and on the other hand you also should wear the gloves, so that nothing should go from your body. Then carefully lay the filter paper onto the membrane, remove the trapped air bubble by rolling the glass rod, cover the whole setup with the saran wrap to avoid the loss of untargeted water, right. Then place a stack of water towel over the filter paper, place the glass plate and weigh about 0.5 to 1 kg on the top of the glass plate. So, ultimately you are going to put very heavy weight, so that all these are actually going to be in connection with each other, and that is how what will happen is that the water will actually evaporate, or water will run from this side and it will reach up to the top. Okay. And when you see that these blotting sheets are wet, then you are supposed to remove that and you are supposed to replace that with the new one, and this you are going to continue for another 16 to 20 hours. Then you allow the capillary transfer for a period of 4 to 16 hours. During this period, if the filter paper get wet, change the filter to maintain the capillary action, because if you do not change the filter paper, then there will be no capillary actions. Do not allow the setup to get dried out. So, you are actually going to cover this whole setup with the help of a saran wrap, and that is how there will be no loss of water except the water what is actually flowing through these filter papers. Once the transfer is done, then you remove the paper towel, remove the membrane and rinse with 2x SSC buffer. Then you check the DNA transfer blotted onto the membrane by visualizing the membrane in a UV light. So, when you visualize in the UV light, you are going to see a same pattern what you have observed onto the agarose, and then you can be able to mark the position of the DNA and different lane with the help of the pencil, right. Then you immobilize the DNA onto the membrane with a UV cross-linker. So, and that is with an optional setting. Okay. So, this is the one method by which you can be able to transfer the DNA from the agarose to the nitrocellulose membrane. The other method is called as the vacuum transfer method. So, in a vacuum transfer method, you are going to use the vacuum. So, compared to the capillary transfer method, the vacuum transfer method is more efficient. Complete transfer of DNA onto the nitrocellulose take place in couple of hours compared to the 16 to 20 hours into the capillary transfer, and capillary transfer is also very slow. So, it actually sometimes causes the diffusion of the bands, so that you are going to have the lower resolution of the bands what you are going to get onto the nitrocellulose membrane. And apart from that, since it is taking very long time, sometimes there will be a degradation of the cut fragments as well. So, treat the gel as described for the capillary method, and then you cut the nylon membrane and the silicone mask with the opening smaller than the size of the gel. So, in this case what you have is you are going to have a tank, and then you are actually going to have the support system, and on this support system you are going to put the your agarose gel, and then you are going to put the nitrocellulose membrane, and then on top you are going to have the vacuum manifold, and then you are actually going to apply the vacuum. So, when you apply the vacuum, the water is actually going to flow from this direction to this direction, and when the water will flow from this direction to this direction, it will also going to transfer the DNA onto the nitrocellulose membrane. And since this is actually being done under the high vacuum, it is going to be much faster than the capillary transfer method. Then you place the wet 3 mm water filter paper on the metal grid, place the nylon membrane on top of the 3 mm water filter paper, and subsequently put the mask. Once you put the mask, then you position the gel onto the mask and place the cover of the vacuum bolt to hold the mask. So, you are going to put the whole bolts on top of the machine, right, and that is how the whole machinery is going to be intact, right. And then you are going to apply the vacuum of 75 mm mercury and initiate the transfer buffer. Then you pour 5 to 10 ml of deep irrigation solution to cover the gel and let the solution permeate the gel, and that is how you are actually going to have the transfer. See the transfer of the gel from the gel to the remove the deep irrigation solution and replace it with a 20 ml denaturation solution and let the solution permeate to the gel. replace the denaturation solution with the neutralization solution and let the solution permeate to gel. Then replace the neutralization solution with 10x SSC buffer and let the solution permeate through gel 30 to 40 minutes. You remove the excess buffer and turn off the vacuums, remove the gel and check the transfer under the UV. Then store the wrapped membrane at the room temperature, and you can actually be able to check whether the transfer happened with the help of visualizing the membrane under the UV lights, and it is actually going to show you the exactly the same pattern what you got onto the agarose gel. Now, next to move to the next step, and next step is the preparation of the probe, right. So, preparation of radio labeled probe. Now, for the preparation of radio labeled probe, you are supposed to know what will be your target gene. Okay. So, if you have the target gene, then you are actually going to identify a region of that particular target gene which is only exclusive, and that particular type of sequence is only exclusive for that particular gene, and that sequence is not present in any other gene. And then what you are going to do is you are going to do a simple PCR, and with the help of the PCR you can be able to prepare the probe. So, this is just a simple example of where you can actually use the random primer method to prepare a radio labeled probe. So, in this method a random primer is used to anneal to the template, and then a PCR reaction is performed in the presence of radio labeled nucleotide. After the PCR, the newly synthesized DNA strand is labeled with the radioactive nucleotide. It has following step. So, what you have is you have first the source DNA, right. So, you have the source DNA, right. This is the region which is exclusive to this particular gene. So, what you are going to do is you are going to have the denaturation of this particular sequence, and then you are actually going to provide the random nucleotides. So, the source strand is denatured to generate the single stranded DNA templates. A random DNA primer is added and allow it to the anneal to the template strand. It will anneal to the random position throughout the sequence at the multiple places. So, it is this random primer is going to add to the multiple position within the template, and then you are actually going to do a PCR with the help of the Cluno fragment and the 4 nucleotides. Now, when you are actually going to add the 4 nucleotides, so primer will anneal to the template strand, and now Cluno will start the synthesis of the new strand, and newly synthesized DNA will give the short stretches of multiple labeled DNA probes, right. So, you are going to have the labeled probe because you are using the 4 DNTPs, but one of those DNTPs are actually going to be radio labeled. So, wherever that DNTP is going to be incorporated, you are actually going to have the synthesized DNA, that synthesized DNA will be radio labeled, and these radio labeled are actually going to be available for using for the into the sudden blotting. Then in the next step you are going to have the hybridization and the development. So, you place the membrane in a hybridization tube and pre-hybridize in 10 to 20 ml hybridization solution for 65 degrees Celsius for at least 30 minutes. So, that is going to ensure that there will be no non-specific reactions. Okay. So, hybridization tube is a kind of a tube, right, where you are actually going to have a cap on top of this, and in this you are actually going to place your nitrocellulose membrane. So, you are going to place the nitrocellulose membrane like this, and then you are going to place your probe along with the hybridization buffers and all those kind of thing, and then this has to be kept in the hybridization chamber. So, in the hybridization chamber it actually rotates in a clockwise direction like this, right. So, it is going to rotate like this, and so that the buffer and the probe whatever you have added should be keep interacting with the membrane. Okay. So, you denature the probe by boiling for 30 minutes and quench onto the ice. Okay. So, before you start the hybridization, you are actually going to denature the probe, so that it is going to be present as a single stranded DNA, and then you are actually going, if you heat it at high temperature and then if you cool it on a ice, then it is actually going to remain as single stranded DNA. Add the probe to the hybridization solution and incubate overnight. Use high temperature for high stringency or the moderate temperature for low stringency. So, depending upon the non-specific interactions, depending upon the pattern of the DNA fragments after the sudden blotting, or the DNA bands after the southern blotting, you can be able to either use the more stringent in reactions, such as you can actually be able to increase the temperature, or you can be able to decrease the temperature. So, that in this particular step you can actually be able to increase the stringency. Then you rinse the membrane thoroughly with the 10 ml of the washing solution and 2x containing 0.1 STS, and expose the membrane to a X-ray film with a intensified screen. And because of this, what will happen is that the DNA is going to be developed. Now, let us see how the southern blot look like. So, this is the southern blot of the DNA of the plant product. What you would see is this is the untreated and this is the treated sample, and what you see here is that you are going to have a very intense band of the fragment of that particular probe, that clearly says that it is actually present as a single copy of that particular gene into this particular plant. Okay, and and on the other hand, what you see also is that it is also present into the untreated sample, but the level of this is very low compared to this, right. So, it also says that this is a single copy of that particular gene which is present into the genome, and the level is also high when you are going to be a particular treatment. So, with this kind of result, what you are going to get in the southern blotting. So, southern blotting, at the end of the southern blotting, what you are going to do is you are going to see a location of the DNA or location of the gene where it is present, because you can actually be able to get whether that particular gene is present onto the chromosome number 3, 4, 5, 6, 7, whatever, and then it also going to say how many copy number, how many copies are present. For example, few genes are actually the single copy number, so they will be only present on the chromosome number 2, they will not be present anywhere else. But there are genes which are also present in the multiple chromosomes, so they will be multiple copy number. Copy number means how many number of gene fragments are present into the genome. So, for some genes they are not been required for running the physiology, or they are very specific, so there only one copy is present. Whereas, there are genes which are housekeeping genes, and most of the housekeeping genes are having the multiple copies present into the genome, so that their production should not be compromised. So, this is all about the blotting techniques what we have discussed, and we have discussed about southern blotting, and southern blotting is a multi-step process which is very lengthy also, in which in the step 1 you are supposed to isolate the genomic DNA, then you are going to digest the genomic DNA with the suitable restriction enzyme or the mechanical sharing, so that you are going to have the multiple fragments, and then you are going to have the you are going to resolve these fragments onto a agarose gel, and then you are going to transfer the agarose gel fragment onto the agarose gel to the nitrocellulose membrane, and you have the two different method what you can use. You can use the capillary method, or you can actually use the vacuum method. Once it is being transferred, then you are going to put them into the hybridization buffer, and you are going to incubate that with the radio labeled probe. The probe is also going to be prepared from a fragment which is very specific to that particular gene, and then after the hybridization you are going to develop the probe with a radioactivity, and at the end you are going to see a pattern where you are going to see the multiple types of bands. So, if the single band is present, this means the single copy is present. If you are going to have the multiple bands, then you are going to have the multiple copies of that particular gene fragment present in the genome. So, this is all about southern blotting. In our subsequent lecture, we are going to discuss about the northern blotting and all other blotting techniques in the subsequent lectures. So, with this I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspects related to blotting techniques. Thank you.